name is Caroline Nelson. This is a presentation in collaboration with Jessica Speckman under Dr. Patel on the regulation of glucose metabolism with the flavanone naringenin in type 2 diabetes and breast cancer. Breast cancer and type 2 diabetes are among the most common diseases. Breast cancer is the second most deadly cancer and impacts 1 in every 8 women. Diabetes is the 7th leading cause of death worldwide. In both of these diseases, glucose metabolism is dysregulated. Naringenin, a flavanone in grapefruit, has been found to impact glucose metabolism in both cell types. In breast cancer, naringenin has been found to decrease the viability and proliferation of the cells. In type 2 diabetes, naringenin regulates metabolism and decreases the accumulation of fat, or lipid, within the cells. While naringenin has cytotoxic effects in cancer cells, in adipocytes it is beneficial and is found to prevent insulin resistance.
In glycolysis, glucose will enter the cell via the insulin-activated transport protein GLUT4 and turn into pyruvate. Pyruvate can metabolize aerobically with PDC, or pyruvate dehydrogenase complex, or it can metabolize anaerobically via lactate fermentation using the enzyme LDH, or lactate dehydrogenase. Adipocytes in individuals with type 2 diabetes have a resistance to insulin which affects the GLUT4 protein. The Warburg effect can be seen in cancer cells where there is an acceleration of glycolysis to support the cell's rapid proliferation. The large glucose intake metabolizes both aerobically and anaerobically to produce ATP. Naringenin has been found to increase lactate production in both breast cancer cells and adipocytes, and we hypothesize that naringenin induces anaerobic metabolism in adipocytes and hinders the Warburg effect in breast cancer cells. In normal adipocytes, insulin is released and binds with the receptor, activating a signal cascade which triggers the GLUT4 protein to allow glucose into the cell. The glucose goes through glycolysis and, without metabolizing, turns into lipid via lipogenesis to be stored. With type 2 diabetes, a large lipid buildup causes a dysfunctionality of the insulin receptor. Naringenin has shown to decrease this lipid buildup, and we are testing the different pathways by observing protein levels to help determine the physiology. These proteins are GLUT4, the pyruvate dehydrogenase complex, and lactate dehydrogenase. The cell samples were categorized by growth, having 3 trials in each group, day 4 and day 16. There was a visual decrease of LDH in the naringenin-treated adipocytes and vehicle control in both day 4 and day 16 trials. However, the comparative difference is statistically insignificant. The PDC trials visually showed a decrease of PDC in day 4 and an increase in day 16. Similar to the LDH trials, however, they showed statistical insignificance. As the gray graph indicates, the presence of GLUT4 in the adipocytes shows a decrease in the treated cells and vehicle control. However, this decrease is statistically insignificant. The breast cancer cells, color green, shows a decrease in GLUT4 for the naringenin-treated cells. MG132 inhibits proteasome, a protein complex that breaks down proteins. The level of GLUT4 with proteasome inhibited can be seen in the third column. When cells were co-treated with naringenin and MG132, the levels of GLUT4 were close to the original control. This indicates that, even though MG132 is inhibiting the breakdown of GLUT4, the presence of naringenin is causing GLUT4 levels to decrease through a different mechanism.
In breast cancer, the expression of the estrogen-related receptor alpha, ERR alpha, has been found to be upregulated. This protein is an orphan-nuclear receptor that acts as a transcription factor to promote the Warburg phenotype. The majority of breast cancer cells are also estrogen receptor alpha, or ER alpha positive, which is a transcription factor that promotes the growth and proliferation of cancer cells. ER alpha is activated by estrogen binding and phosphorylation. The increased presence of ERR alpha and ER alpha is highly correlated with tumorigenesis. Naringenin has been found to regulate both of these transcription factors as well as GLUT4 in cancer cells. Our experiments found that naringenin decreases the expression of both ERR alpha and ER alpha protein levels in MCF7 breast cancer cells. MG132 is a proteasome inhibitor. The co-treatment of naringenin and MG132 suggests that naringenin does not produce its effects through labeling the proteins to be degraded in proteasomes. Naringenin, by decreasing the expression of these key transcriptional factors, may be causing a change in the metabolic profile of these cells to inhibit the Warburg effect. Through this mechanism, it is hypothesized that naringenin may be able to prevent tumorigenesis. In conclusion, naringenin has been found to regulate the expression of several key proteins in breast cancer cells and adipocytes. Further experimentation is needed to determine if the flavanone can be used therapeutically, though the current results are promising. Our next steps will be to perform a lactate assay to determine the lactate levels in adipocyte cell lysates. It is important to note that the results of our experiments have been found to be congruent with literature.